The evolutionary adaptation of Escherichia coli's ability to grow at suboptimal temperatures. The global mean temperature has already risen by 1 degree Celsius since 1850, and with current rates we could expect to see another 1.8 to 4 degrees Celsius increase before the end of the century. There has already been extensive research done to see the effects of global warming on larger scale organisms. We wanted to determine if this substantial increase in temperature would have an effect on environmentally important microorganisms such as E. coli. Research suggests that E. coli can withstand temperatures between 32 and 43 degrees Celsius, with 37 degrees Celsius being the optimum and 43 degrees Celsius being the tail end of temperature tolerance, depending on the strain of E. coli. The purpose of this project was to see if E. coli has the ability to adapt to suboptimal temperatures. Based on research, we hypothesized that E. coli would be able to rapidly adapt to different temperatures.
To start our experiment, we collected water from 2 different habitats, the North River in Bridgewater, Virginia, and Black's Run River in Harrisonburg, Virginia. We isolated E. coli through the Colliskin filtration method. The filtered E. coli was placed on a petri dish and incubated for 24 hours. Next, we transferred colonies from the filter to an EMB plate and then to a TSB tube. The E. coli was placed in different TSB tubes at 25, 30, 37, 39.5, and 42 degrees Celsius. 3 replicates were grown at each temperature. Once a week for 3 weeks, a growth curve was conducted of the E. coli over a 12-hour period. The absorbances were read from a spectrophotometer at 600 nanometers, and calculations were performed to analyze if any adaptations have occurred.
This figure depicts the results of the samples from the North River. After 1 week, the tubes incubated at higher temperatures had higher growth rates, and those incubated at lower temperatures had lower growth rates compared to the control. The 25 and 30 degree strains did not experience much change in their growth rates over the 3-week period. The 39.5 and 42 degree strains decreased their growth rates in week 2, and the 39.5 degree strain increased its rate again by week 3, indicating potential adaptation. The second figure depicts the results of the samples from Black's Run. Similar trends are seen in week 1 as those seen from the North River. By week 3, the 42 degree strain had increased its growth rate again, similar to the 39.5 degree strain from the North River. Due to the time constraints, we had to reject our hypothesis because it seems a 3-week time period was not sufficient enough time to see temperature optimum shifts from 37 degrees. We were able to determine that E. coli can withstand the temperature increase caused by global warming. However, we were unable to determine how long it would take for E. coli to adapt to new temperature conditions.